Forensic Science Lecture 10-3, Polymerase Chain Reactions and Short Tandem Repeats. The most important information that the PCR process gives us is about the DNA polymerase, that it can be directed to copy a region of DNA. To perform PCR, short sequences need to be identified first, and they need to be available in their pure form. These pure forms are known as primers. The process of PCR first starts when the DNA is heated to 94 degrees Celsius, and this causes the double strands to separate completely. The second step is to add the primers and lower the temperature to 60 degrees Celsius. This allows for the primers to bind to the strand, which is now known as hybridized. And so the primers tell the DNA polymerase where to begin. The third step is to add the DNA polymerase along with free nucleotides. It's then heated again to 72 degrees Celsius, and this allows the DNA polymerase to build the new strand. This is one complete cycle. Typically, there's anywhere from 28 to 32 cycles that are carried out. That can create more than 1 billion copies. Each cycle takes less than 2 minutes to complete. The advantages of this is that it is best applied to strands that are no more than a couple hundred bases long. The shorter the strands, the more stable, and they're subject to less degradation from the environment that they're in. They can be amplified from minute amount of DNA, so this overcomes the limited size sample problem that investigators had come across. And they're extraordinarily sensitive, so it allows for small quantities to be characterized. Whereas with the RFLPs that we talked about earlier, or the tandem repeats, they are too long for the PCR process. The latest method of DNA analysis actually uses what's known as short tandem repeats, or STRs. And it is actually the most successful and widely used to date. These short tandem repeats are significantly shorter than the RFL tandem repeats. With the STR, there is little as 100 and 125 picograms, whereas 100 times, that makes them 100 times less than what's normally required for the RFLP analysis. They are short tandem repeats, or STRs, are located on chromosomes, or locations on chromosomes that contain short sequences that repeat themselves. So when we say short, the sequences contain 3 to 7 bases as opposed to 15 to 25, which is again much less susceptible to degradation and often recovered from stains or bodies in extreme decomposition. So we have the ability to obtain these more easily than the longer strands. Because they are so short, they're ideal for the PCR process, again overcoming the sample size limitations. One common STR used is known as the TH01. This is a repeating sequence of AATG. There are 7 variations of the TH01 STR that have been identified, and they contain anywhere from 5 to 11 repeats of the AATG. The TH01 is extracted, amplified by PCR, and then ran through electrophoresis. And by examining the distance the STRs have moved, they can determine the number of repeats within that DNA sample. Everyone has two types of TH01 STRs, one from each of their parents. As always, when we talk about examining evidence, multiple pieces of evidence is always better. So the more STR characterized, the smaller the percentage of the population that have that combination. This gives rise to a concept called multiplexing, and it uses PCR technology to simultaneously extract and amplify a combination of different STRs. The forensic community in the US have standardized 13 STRs and entered them into the CODIS database. And because of this, it allows companies to manufacture STR typing kits that can simultaneously make copies of 15 STRs at a time in order to be compared and determine the frequency in that population. So the probability of identity is the likelihood that two individuals would have identical STRs. The smaller the value, the more discriminating the STR. The high degree of discrimination and even individualization can be obtained by multiplexing. The probability that two individuals, or the probability that they use that two individuals would have the same STRs is based on two randomly selected individuals. So more than likely, they're not going to have the same, and so the bigger the difference, or the less likely it is, the more that individual is associated with that sample. We have a need to reduce the analysis time of DNA analysis. And so capillary electrophoresis has been developed. Again, they can still be developed through the gel-coated electrophoresis, but it takes more time. The capillary electrophoresis is a process carried out in a thin glass tube rather than the gel coating plate. Ends of the column are submerged in buffer with electrodes. The tube's coated with gel, and the sample is injected into it. Fragments are going to move through the capillary or the tube to a detector that's then going to record the data. So the information is the same, just takes a little less time. One thing that the STRs can give us is sex identification. And so the STR kits have the ability to identify what the sex of that DNA contributor is. And it focused typically on the amygdaline gene, and this is located on the X and Y chromosome. So it's a sex-linked gene, but it actually is the gene for tooth pulp. It's actually 6 bases shorter on the X chromosome than it is on the Y chromosome. So that is one gene that they'll focus on to see if it's a male or a female. Another STR analysis is the Y chromosome analysis. So the Y chromosome, remember, is solely for maleness. It develops maleness in male individuals. And so they've been able to identify more than 20 Y STR markers. And so when they're testing for these markers, especially from like a vaginal swab, the female DNA that's present doesn't show up in the sample because there's no Y chromosome to identify those markers. Traditionally, also, the STR markers from a vaginal swab of the male donor only last 3 to 4 days. But when looking for the Y STR markers, that extends that time to 5 days. So it kind of gives them a better chance of identifying who that assailant is. The significance of DNA typing, especially STR DNA typing, has become essential and a basic tool for law enforcement. DNA evidence is impartial in indicating both guilt or exonerating the innocent, and so it is uh, a highly looked at piece of evidence. Um, it also, on the other hand, has placed an added burden onto crime labs, which isn't necessarily a bad thing because it does force them to improve their quality assurance procedures and ensure correctness of the results. So it's more accurate in identifying um, who is involved and who isn't involved, to ensure the right um, verdict in the trial. This ends lecture 10-3. There is a quiz next class.